The cephalopod's body plans and habits are so very different from those of humans that they might almost be aliens from another world. So perhaps it's not surprising that it took a long time for scientists to discover that there are fundamental similarities between the nervous systems of cephalopods and vertebrates. Yet it was the recognition of a useful difference in their nervous system which enabled scientists to undertake research that has led to a growing understanding of the mechanisms controlling our own nervous system. The breakthrough concerned the nerves that control the contraction of the mantle muscles used in jet propulsion. As this archive film shows, by simultaneously contracting its mantle muscles, even a moderately sized squid can eject a huge amount of water with great force. In the mid-1930s, the British zoologist Professor J.Z. Young was engaged in a study of the squid's anatomy. Young observed an array of large tubular structures, each as much as a millimeter in diameter, in the squid's mantle. As these structures were never filled with blood, they could not have been blood vessels. From their similarity to surrounding nerve fibers, Young thought they must be single neurons, giant axons, that transmitted nerve impulses from a concentration of nervous tissue, called a stellate ganglion, to the mantle muscles. Using electrodes, he stimulated the surrounding nerve fibers and found that he could only produce large muscle contractions in the mantle when the large tubular structures remained intact. So, these were indeed giant axons. Scientists quickly appreciated the significance of Young's finding, for here at last was an axon large and robust enough to investigate with the techniques available at the time, and one that survived for several hours when isolated from the nucleus. The intracellular contents of the giant axon could be removed and analyzed, leading to the discovery that sodium ions were more concentrated outside the nerve cell and potassium ions more concentrated inside. By refilling the empty axons with solutions of precisely known chemical composition, experimenters were able to unravel the mechanisms of ion transport across the membrane. The giant axons are large enough and robust enough for fine electrodes to be inserted through the cell membrane and into the axoplasm. In these early techniques, a fine glass tube was first inserted into the axon and secured with thread. Then the tube was used to introduce a fine wire electrode from which the voltage between the inside and the outside could be measured. But the formation of the nerve impulse was far too rapid for detailed study with any of the electrical measuring devices of the late 1930s. It wasn't until the 1950s, following the wartime improvement of electronic equipment such as the cathode ray oscilloscope, that major progress was made. Scientists found that the nerve impulse was transmitted as a characteristic wave of electrical potential, and that this all-or-nothing action potential was generated mainly by transient movements of sodium and potassium ions across the nerve membrane. Research on the squid giant axon unraveled the mechanisms of the formation and propagation of the nerve action potential. This understanding led directly to the development of drugs that block action potential formation and so act as local anesthetics, now used routinely as painkillers in dentistry and minor surgery.